My name is Nikolaus Fortelny and I'm a group lead at the University of Salzburg in Austria. I will be talking today about reliable interpretability of deep learning on biological networks. And to explain what I mean here is one example of such biology-inspired deep learning or deep learning on biological networks. And this is the well-known PNET model, which was published in 2021. And the idea is that this is a deep learning model, but the architecture of the computational graph of the network is based on prior biological knowledge. So this specific model, they used mutation data linked to genes. Mutations are in genes, linked to genes. The genes are grouped in pathways and biological processes, and this is based on annotations in biological networks to predict. And what was predicted in this model is different cancer phenotypes, most importantly metastasis, so whether cancers metastasize or not. In pancreatic cancer was this particular example. So in this case, the neural network was based on the Reactome database. The input was mutation data and the output patient metastasis. By having such a network, where the network is based on biological knowledge, one can interpret the individual nodes in the network as they are sparsely connected and have meaning. So when we want to get those interpretations, we get importance scores for the hidden nodes, and these provide hypothesis for follow-up experiment, for example, which pathway may be involved in metastasis. These pathways themselves are not directly measured, they just provide interpretation of these results. As a second example of biologically inspired models, I previously developed so-called knowledge-primed neural networks. These networks are slightly different. The input here is not mutation but single-cell expression data. The output is whether cells were stimulated or not, and the network is based on the signaling database, SignalDB. So the network here consists of kinases and transcription factors and receptors. So it's really a model of signaling in the cell, so to say. Then when we interpret the hidden nodes in this network, we get interpretations that are protein activities here. Doing this analysis, I found two key points that are relevant and required to enable good interpretations. So one is to control robustness of interpretations, and the second one is to correct for network biases. And so I'll now explain what this means. So robustness means that if I train the same model 2 times, do I get the same results? So robustness can also mean that we just have small variation in the data and do we get the same results, but here really mean same data, same network, same output, but do we get the same results? Here, shown on the left, is one figure from our paper showing the simulated network where there are 2 hidden nodes which are connected to informative genes, and the other ones are not informative genes. So those 2 get higher scores, but in a standard approach, they are inversely correlated. So if one gets high weights, the other one doesn't, and vice versa. And this means that here those are replicate networks, and if you train one, the results may be very different when you train a second. Now, when we use dropout here, so dropout is an algorithm from machine learning, then you will find that those nodes, those weights are much more correlated, and this somehow improved the robustness, but still we found that we had to train multiple networks. A second thing we discovered in KPNNs were network biases. Network biases means that not all nodes have the same number of connections. This is very well known for biological networks with hubs and so on. And so here we created another simulated network where this node has less connections than this one, and you can see it also gets less weights. To approach this problem, we developed ways of training on control data. Control data means that in this case all genes had the same level of predictiveness. So when we get interpretations for these nodes from control data, then these interpretations somehow reflect only the network biases, while when we train on real data, so this would be an example of real data, a simulated example, then we have data and biases. And by then comparing the two, data versus the realistic data versus the control inputs, we get more meaningful and relevant interpretations. So these were developed in KPNNs, but they have not been studied in other biology-inspired models, and there are quite a few at this point. This was published in 2021, and you can imagine this is a very interesting field. So our question was, do these issues, robustness and network biases, always also affect other models? And we wanted to test this in this prominent example of the peanut model. So the first experiment we did was to test the robustness. So just to again iterate this idea, we have a network at the beginning, and it creates certain initial weights. So initial weights in deep learning are randomly picked. And then it trains and it gets certain final weights and importance scores and interpretations. Now, when we get a second network or a third network, it has different initial random weights because these are random, they are meaningless, and it may get slightly different or more different importance scores. And the question is, how similar are those importance scores at the end based on these different initial weights, which are again meaningless? And we do find that these indeed affect interpretations. So what you see here are in the different layers of peanut, the nodes, and you see that the black box plot is showing the interpretations from training many times, and the red one is the interpretation of the original model. So in this publication, only one model was trained, this is the red dot, but we now trained multiple times producing the black box plots. And you can see that in some cases, the interpretations are really very different. And in some cases, even the first and the second most important interpretation is switched. So we can see that this is one example comparing two, so the original network and one replicate network, and you can see that the order of nodes can be switched quite dramatically based on this. So this shows that the importance scores are affected by repeated training also in this case. Now for the second part, we wanted to test whether these network biases affect interpretations, and we had two approaches. One again is the so-called deterministic inputs. So we call them deterministic because they're perfectly predictive of the output. So you can see here the mutations and the CNVs are perfect. So here we have, let's say, ones, and here zeros, and the labels are also 1, 0, and 0. So they're perfectly predictive, and this is a control approach. We used a second control approach where we randomly shuffled the labels. So in the original model, there is some predictive power, but when we randomly shuffled labels, there is none. And so we tried both of those. And so doing that, we were able to see that again, our control inputs, both of them, the deterministic inputs as well as the random labels, produced importance scores. Interestingly, they were, when doing this multiple times, they were correlated, meaning that these are meaningful importance scores and not just some random values. And we could compare them to the original values. So in black, in both cases, you see the original importance scores for the different layers. And you can see here that for some nodes, the original importance score is higher than the controls, but for some nodes, you can see also that the controls are higher than the original importance scores. So again, this enables us to compare the importance scores obtained from real data to our controls and see where the network biases exceed what we have seen before, and enables us to control this. Finally, wanted to understand which network biases are driving these interpretations. And so we tested the correlation of our three setups, so the original setup, the deterministic inputs, the control, and the shuffle labels as a control, and correlated them with different measures of node centrality. And we found that specifically global measures of centrality, such as reachability and betweenness, were highly correlated with node importance scores in the original setup and in also the shuffled one. All right, so the conclusions are initial weights affect interpretations, we need robustness, network properties affect interpretations, we need to control for them. This has been observed in KPNNs and Peanut and probably affects all other models. And so we need to repeatedly train and think of the right controls. With this, I would like to thank you. And these are pictures of my team. I specifically want to show Wolfer Extrascala here, who was driving this research. Thank you.